Chapter 13, GI System. The GI tract is the body's point of entry for nutrients, including fluids and electrolytes, needed to sustain life. It is also the route of most medications that are administered. The GI tract is complex. It functions as the gateway to oral nutrient, drug, and toxicant access to the rest of the body. It is the most critical site of fluid and electrolyte homeostasis outside of the kidneys. It is the site of digestion and absorption of nutrients. It is the route of excretion of insoluble dietary fiber, products of liver transformation of endogenous compounds and xenobiotics, and cholesterol. It is the body's largest immune organ, with cells and tissues of both innate and adaptive immunity. It is the only organ with a freestanding nervous system, the enteric nervous system. It has a high concentration and variety of neurotransmitters, modulators, and hormones, greater than most other organs outside of the brain. And its mucosal epithelial cells have a high turnover rate, potentially removing cells that have sustained damage. But also it is associated with vulnerability to malignant transformation. Disorders of the GI tract are often grouped into the following categories: alteration of the digestive function, absorptive function, immunologic function, and neuroendocrine function. Many acute GI disorders result from infections due to bacteria, virus, or parasites. Chronic inflammatory bowel diseases such as Crohn's disease and ulcerative colitis cause significant disability, including intestinal failure. Chronic GI disorders also may present with pain, diarrhea, or constipation, as well as other signs and symptoms of malabsorption and critical nutrient deficiencies. These are various GI disorders for the pediatric individual and the gerontologic individual. The major function of the gut is to mechanically and chemically process materials or digest for absorption. The process of chemical absorption depends on hydrolysis, or the breaking down of dietary macromolecules into their components. Starches break down into monosaccharides or sugars. Proteins break down into peptides and amino acids. Lipids break down into fatty acids, monoglycerides, and cholesterol. This breakdown is followed by transport of these components across interstitial epithelial cells that line the lumen. Accessory organs are salivary glands, the liver and gallbladder, and the pancreas. These all supply digestive fluids such as saliva, bile, and digestive enzymes. This is the general structure of the GI tract, components that surround the lumen. The mucosa is the layer of epithelial cells over connective tissue and a thin muscle layer. The submucosa contains blood vessels, glands, lymphoid tissues, and a nerve plexus. The muscularis are layers of circular and longitudinal smooth muscle separated by blood and lymph vessels and a nerve plexus. The serosa is connective tissue and has a covering of an epithelial layer. Neurons in the two plexi of the enteric nervous system have sensory and motor connections with the autonomic nervous system. The parasympathetic efferent supply comes from the brainstem dorsal motor nucleus of the vagus and the sacral parasympathetic nuclei. The vagal afferents project into the brainstem. The sympathetic supply comes from preganglionic neurons in the thoracic spinal cord that rely on the sympathetic ganglia, accompanied by spinal afferent fibers. The autonomic control of the gut by the parasympathetic neurons of the vagus nerve and the sympathetic neurons from the spinal cord. The vagal parasympathetic neurons are shown in blue, are located in the dorsal motor nucleus of the vagus nerve, with efferent fibers carried by the vagus nerve or cranial nerve 10, and it is identified by an X in the figure. The gut afferents are sensory fibers indicated by projections from the GI tract back to the brain, relay sensations of stretch, pressure, irritation, and pain via the autonomic nerves. The vagal sensory fibers terminate in the nucleus of the tractus solitarius. The neurons of the ventrolateral medulla regulate the activity of the spinal sympathetic neurons shown in red. These also receive spinal sensory inputs. Within the brainstem, the area post-trema and the NTS modulate both parasympathetic and sympathetic outflows. Higher centers, particularly limbic cortex, interact via multiple pathways that project through the amygdala to the hypothalamus to influence GI activity through the autonomic nerves. These centers, shown as purple, contribute to stress-induced alterations in GI signaling and disorders. Gut hormones allow signaling from one segment of the GI tract to distant segments, gallbladder, and the pancreas, to coordinate response in successive phases of meal digestion and absorption. The endogenous opioids regulate GI function through the opioid receptors. Their actions are inhibitory, thus promoting constipation. Exogenous opioids used for pain management often cause severe constipation. Stimulation of gut opioid receptors inhibits gut neurotransmission, motility, and secretion. Therefore, morphine and other opioid drugs used for pain management or taken as drugs of abuse have significant side effect of constipation. These gut microbiota are usually non-pathogenic and contribute to normal gut function. To protect against overgrowth of normal microbiota, as well as ingested pathogens, the gut epithelial layer and submucosa have extensive immune surveillance and effector functions, including antimicrobial secreting Paneth cells, plasma cells that secrete IgA, colonic goblet cells that secrete mucin and protective proteins, M cells that take up antigens and interact with immune cell clusters in the submucosa, gut-associated lymphoid tissues like Peyer's patches, and freely moving DCs, macrophages, as well as T and B lymphocytes. The immune cells, the B cells, T cells, macrophages, and dendritic cells, are distributed in many gut-associated lymphoid tissues. Clusters of lymph cells called Peyer's patches in the lamina propria beneath the epithelial cell layer. Peyer's patches are found throughout the small intestine and are particularly numerous in the ileum. In the epithelial layer, the cells contributing to immunity include intraepithelial lymphocytes, Paneth cells, and M cells. Esophageal motility consists of peristalsis of the bolus of swallowed food. Esophageal obstruction is typically caused by mechanical stenosis from inflammation, by chronic gastroesophageal reflux or GERD. Achalasia is a condition of esophageal obstruction due to the loss of inhibitory neuron function. Esophageal varices are dilated veins in the esophageal submucosa that are the result of a port-caval shunt secondary to portal hypertension. With this additional load of blood flow through small veins, the varices can bleed profusely if they are ruptured. GERD esophagitis initially triggers hyperemia with ongoing inflammation leading to leukocyte recruitment and infiltration. Chronic GERD may lead to a change in the esophageal epithelium from stratified squamous epithelium to simple columnar epithelium. This is known as Barrett's esophagus. Adenocarcinoma typically originates in the distal third of the esophagus and is often associated with Barrett's esophagus. The factors contributing to GERD can include dysfunction of esophageal motility, decreased integrity of the lower esophageal sphincter, decreased intrathoracic pressure, increased gastric pressure, and increased gastric acid production. The stomach is the most distensible and muscular part of the GI tract. It stores and grinds food, initiates protein digestion, kills bacteria with gastric acid, and dispenses food particles into the small intestine as chyme. In the fundus and the body of the stomach, the gastric pits are dominated by three main cell types: mucous neck cells, chief cells, and parietal cells. Activated parietal cells expend large amounts of adenosine triphosphate on active transport by the proton pump. This transport moves hydrogen ions into the stomach lumen in exchange for potassium ions. The parietal cells secrete acid. As food intake begins and throughout a meal, the vagus nerve releases acetylcholine, stimulating parietal cell muscarinic receptors via the GQ and initiating acid secretion. The parietal cells also secrete the intrinsic factor, which is a glycoprotein carrier for the uptake of vitamin B12 via receptors located in the ileum. Parietal cell acid secretion is mediated by the active transporter hydrogen potassium ATPase, also known as the proton pump. The chloride ion moves to the stomach lumen through the ion channel. The proton pump activity is strongly regulated by neural, endocrine, and paracrine signals acting on the G protein coupled receptors. The major factors stimulating acid secretion are acetylcholine, gastrin, and histamine. All of these act through GQ and GS. Prostaglandin E2 inhibits the proton pump through a G-linked receptor. Pharmacological targets of H2 blocking drugs and proton pump inhibitors are shown. Each of these three phases secrete stomach acid depending on whether food is present or someone is thinking about food. There are several protective factors to help maintain gastric integrity against acid and enzyme-induced damage. The mucus cells produce adherent mucus, which is bicarbonate-rich, in response to prostaglandin stimulation. The gastric epithelial cells are held together by tight junctions to minimize the acid diffusion to the submucosa. These cells also undergo rapid turnover, quickly replacing damaged cells. The gastric mucosal blood flow is high and maintained at that level by local prostaglandin production. This high rate of blood flow quickly removes any acid that leaks through the tight junction barrier and promotes repair of damaged tissue. Peptic ulcer formation is erosion of the mucous membrane of the stomach, or more commonly, the duodenum, and it can stem from many causes that lead to an imbalance between damaging and protective forces. Stomach acid and pepsin can directly damage the mucosa. Stress can assist to trigger the inflammatory process and enhance histamine release. The increased sympathetic activity can reduce the blood flow. Non-steroidal anti-inflammatory drugs, or NSAIDs, inhibit prostaglandin production. These reduce the parietal cell inhibition and gastric blood flow. The single greatest risk factor for ulceration is the activity of the gram-negative bacterium H. pylori. It is unique in its ability to survive in acidic environments. This is partially due to its production of the enzyme urase, which converts urea to ammonia to neutralize gastric acid, thereby allowing a local buffering effect. The cytotoxins released by H. pylori then break down the mucosal cells, causing gastritis. Histamine from the subsequent inflammatory response increases the acid production. Management strategies for ulcers must eradicate the H. pylori infection to prevent ulcer reoccurrence. Gastric motility consists of fundic relaxation early in meal consumption and strong phasic and tonic contraction of the corpus or the body and antrum to pulverize meal contents while promoting mixing with gastric secretions. Pacemaker cells set the frequency of gastric smooth muscle activity. Antral contractions move chyme to and through the pyloric valve into the duodenum. Gastroparesis is a state of decreased stomach motility that results in slower gastric emptying, nausea, vomiting, abdominal pain, and premature satiety. The most common causes are diabetic, idiopathic, and post-surgical. Vomiting requires coordinated activation of abdominal muscles, diaphragmatic contraction, and gut smooth muscles. Neurotransmitters involved in vomiting initiation include central or peripheral serotonin and dopamine, substance P acting on neurokinin type 1 receptors, muscarinic acetylcholine receptors, and histamine type 1 receptors. These are the targets of the different classes of antiemetic drugs.